My name is Guo-Ping Shi from Boston, Brigham Women's Hospital. I'm associate professor of Harvard Medical School. I just had my presentation here at the 22nd International Congress of Heart Disease here in Vancouver. I just talk about our recent work about sodium proton exchanger proteins in human and murine atherosclerosis. My research have been focused on several things in the past number of years, such as asthma, mast cells, and IgEs in this cardiovascular disease. One important thing that we found is that IgE, for example, activates mast cells and promotes atherosclerosis. One novel finding that we recently discovered is that IgE can induce macrophage apoptosis. One of the mechanisms is by reduced extracellular pH by activating a protein called sodium proton exchanger, called NHE1. As a result, the pH gets dropped because the proton pumped out to the extracellular matrix and the extracellular space. And when the pH drop, the whole cell, extracellular medium getting like acidified. So we proposed the hypothesis that in human atherosclerotic lesions or in mouse lesions, the area was rich of macrophages should be acidic. So we tried to propose a study to determine the role of these proton molecules in atherosclerosis. And we found that if you knock out the genes, you can reduce atherosclerosis. And the most important, I think, novel finding, interesting finding, is that we found that the human lesions that in area with lots of macrophages or undergo apoptosis, that area is acidic, has a lower pH. And we proved that's true in both mouse and humans. And we are able to manipulate the systems by deleting that genes in NHE1 and change the pH and prevent atherosclerosis. And as the future applications, we developed a method by giving the mice, like IV injections, a compound. And that compound is high energy and pH sensitive. We used as a probe that can be used to monitor in real time of atherosclerotic lesion development in mice. And when we knock out genes, and actually this probe can, under acidic pH, can release certain number of signals that can be detected by FMT, FMT-CT, to detect the signals in without killing the mice. So when you knock out the genes, we found that the signal can, significantly reduced. So in the future, we think probably similar molecules can be used in humans. We hope that it will be the case in the future. So we can, for example, we can give the compound to the human veins, the blood IV injections. Then once to reach this area with plaque, macrophage apoptosis or necrosis in that area, so there are a lot of, pH changes, and that probe can release signals that can be detected by FMT technique. At least this can be done. Can easily be done in mice, in animal work, and the future is also in animal work without killing the mice. But in humans, we hope that will happen. That will be the case. So overall, this study that I just presented has three components. One is we continued our earlier studies show that IgE involve a atherogenesis as one of the mechanisms is activate NHE1 molecules, which then activate the macrophages. This is the one component. The second conclusion is that when you knock out the genes, you can basically mute the action of IgE and prevent atherogenesis at the molecular cellular levels. And lastly, we found that we can use this observations, develop a probe to monitor human atherosclerotic lesions by without killing the mice, by in vivo real time, and giving the probe to the mice, and wherever has lesions, they can release near-infrared signals that can detect by FMT technique. So this is like a three major components of my talk.